online. So thank you all for coming today to the first CGTG seminar of the year. And we have Christina Rochus, who's a core scientist in Ivan Porshnik's lab and joined the CGTG committee in September.
Yes.
Yes. And so Chrissy will has a research interest that includes genomic data to study genetic diversity and adaptation. She has a BC in animal biology and an MC in integrative biology from the University of Guelph in Canada, and a double PhD in animal breeding and genetics from the Swedish University of Agricultural Sciences and INRAE. And postdoctoral training at Uppsala University, Cornell University, Wageningen University, and the University of Guelph. Prior to coming to the Roslin, Chrissy was a research associate at the Center for Genetic Improvement of Livestock, Department of Animal Bioscience at the University of Guelph in Canada, where she was working on the Resilient Dairy Genome Project. Thank you for being here today, Chrissy, and the floor is yours.
Thanks. Thanks so much for the introduction, and thank you everyone for being here today. So as you can see, I'm going to be presenting work that I did while I was at the University of Guelph. That works. So I'm going to first look a bit broadly, and because this work was part of the Resilient Dairy Genome Project. And so what is resilience? And in the group that I was working in, we were particularly focused on dairy cattle. And so we said a resilient cow is able to adapt to changing conditions. So there are a lot of different definitions of resilience out there, but we were particularly interested in focusing on certain changing conditions and certain traits that we wanted to focus on. So one of those changing conditions is global demand, climate change, and human and animal health. And it's really important that our dairy cattle are able to adapt to these different new environments so that we can continuously have a really good source of protein to feed people. So we were looking at new traits and what can we start to include in our breeding programs that can help with increasing or maintaining the resilience of dairy cattle. So I think a lot of people, when thinking about the environment particularly, probably think about these traits, the more resource efficient and also reduced environmental burden, so decreasing the methane emissions from dairy cattle. So there were people in the group who were looking at these traits. We also had people looking at fertility traits and what we call closer to biology fertility traits, because right now in dairy cattle, a lot of these fertility traits that we use for selection purposes are highly influenced by management practices. And so by getting closer to the biology, we can do a better job of selecting for these new traits. And actually, Audrey was working on this project. But today I'm going to talk mostly about health. And so we looked at a lot of different diseases. And I'm just going to present two of those to you today. And sorry. And so while we were looking at all of these traits, at the same time, we also wanted to make sure that our animals are still highly productive. So as I said, this was part of the Resilient Dairy Genome Project, which is a very large project spread across Canada, but also we had collaborators across the world. And I, in particular, was also funded by MyTax Elevate scholarship, and this was in partnership with Lactanet, and Lactanet Canada does the genetic evaluations for dairy cattle in Canada. So I'm going to talk about Leukosis and Johne's disease today and resistance to those. And I decided to focus on these two in particular because I think in a similar way, they're both really good candidates for novel traits that we can select on. So for both of them, if I can kind of generalize, they're both chronic diseases with no treatments. So once an animal gets infected, they will have this for the rest of their life and could even be fatal in both cases before they're culled. There are no leukosis vaccines, and there are no commercially available vaccines for Johne's disease in Canada. So this also takes that aspect of management away from producers as well. They both have long incubation periods, so your animal could get infected or exposed to another animal right away, and you're not going to know whether they've been infected, and it could be for years. And then finally, subclinical signs are difficult to detect, and you're probably thinking subclinical, yes, you're not supposed to detect those. But actually, some of these signs can include reduction in milk production, poor fertility, weight loss. And so these are things that producers could notice, but they're not necessarily going to go directly to, oh no, my dairy cattle have leukosis or Johne's disease, and they shouldn't. So I think that with these difficulties in management practices, that we can start to think about how can we breed for more resistant animals so that we can make permanent progress in that area. And then hopefully also, well, simultaneously other animal scientists are working on other ways that we can manage these diseases. So both of the projects I'm presenting today, the first one I'm going to present is on leukosis, and then I'll present about Johne's disease, are both projects from master students at the University of Guelph. So this first project was from Renee Bongers, and she was looking at both the prevalence of leukosis in Canada, as well as estimating heritability, and then also doing a GWAS. So the full name for leukosis is enzootic bovine leukosis. I'm just going to stick with leukosis for today. It's a chronic infectious disease caused by the bovine leukemia virus, or BLV, and it's spread through transmission of infected lymphocytes. And so lymphocytes are just a type of white blood cell. They're endemic, so leukosis is endemic to North America, and it's increasing in prevalence. So in the 80s, it was estimated to be about 40% in 40% of herds, and an estimate from 2015 is about 90% of herds. So one of the control strategies is to prevent infection through culling of infected animals. And so you might be wondering at this moment, why do I not know about leukosis? And it's because the last time it was known to be in the UK was in 1996. And this is because countries took different different approaches, and in Europe, typically, there was this culling of infected. And unfortunately, I think now in North America, we're at the kind of level of prevalence that it's not really possible to cull all of those animals that are infected. So it's an interesting contrast between the different strategies that have taken place. So as I said, there's no treatment or vaccine available, and there has been a small study from the US where they found heritability of 8%. So the signs and costs of leukosis, so the subclinical signs, as I said, decreased milk production, impaired immune response, and so a lot of opportunistic infections, especially like mastitis. You'll see a lot more of that in animals that are infected with leukosis. Reduced fertility, so longer number of days open, so more times needed to try and inseminate cattle before they're pregnant. And then poor longevity, and this is probably just tied back to this idea of difficult to get these cows pregnant, and reduced milk production means that they're probably going to get culled early as well. Clinical signs persistent lymphocytosis in 30% of animals infected, and then 5% of animals can lead to this malignant lymphoma. Let's see. So the Dairy Farmers of Canada have estimated that the average test-positive herd in Canada loses about 12,000 to 19,000 Canadian dollars per year, or about 7,000 pounds to 11,000 pounds per year. And this is assuming like 100 animal dairy herd. So our objectives were to examine the prevalence of leukosis in Canada with the records that we have available, to assess the potential for selection for leukosis resistance using genetic parameter estimation and estimated breeding value correlations, to estimate heritability of leukosis resistance incorporating genomic information and predicting genomic estimated breeding values, and perform genome-wide association study to understand the genetic background of leukosis. So we had data from 2007 to 2021 from 950 Canadian herds. We had over 96,000 Holstein cows that we had records for, and over 117,000 milk enzyme-linked immunosorbent assay or ELISA test records. And the handy thing about these kind of tests are that leukosis testing is done through routine milk testing, but it is voluntary and it's an extra cost for producers. So this is why not all herds opt in, and we'll see this more in the results. So for the leukosis tests, there's 3 options basically for the results. You have positive, suspicious, or negative. So we removed all suspicious results because sometimes the sharing of the milk recording equipment can lead to cross-contamination. And then we only kept 1 record. So the first time an animal tests positive, then we just say that's the moment that they were positive. Okay, so we had up to 13 generations of pedigree data. We had over 22,000 animals genotyped or imputed to 50K SNPs. And then we also had data on other traits, production-related traits, but also the indexes that are used in Canada often by producers to select for their next generation. And then we used BLUP F90 program softwares to do our analysis. So we have our animal model that we used, and so our trait was the leukosis test results, so we only had those negative or positive, so binary trait. Then we had our fixed effects of year, season of calving and lactation age at calving. Class effects, we had a random effect of herd year of calving effects, and then our additive genetic effects and our residual. We also tested a threshold model because ideally with binary traits you want to use the threshold model. We did a comparison of them and we found that we got statistically the same results when we compared like the ranking of sires from the EBVs. So I'm just going to show you the animal model because it's simpler and easier to interpret. And then we also did single-step GBLUP. So instead of using an A matrix to represent the pedigree relationships, we use pedigree information as well as SNP data to make the age matrix. Okay, so prevalence of leukosis in Canada. So of the herds that test for leukosis in Canada, it's about, well, I think it was about, it's difficult to see from here. It's about 80% of herds that are testing positive for leukosis of those that test. This is a bit difficult to interpret because typically like especially at the beginning of the testing in 2007, probably producers who were aware that they had leukosis or issues with leukosis were probably the only ones testing. And now that we're, it's like 2020, that you can see that there's actually more herds that are being tested, but still a similar amount of herds that are testing positive. So it's probably just that farmers are more aware now about leukosis and more farmers are opting in for testing. Yes, and then there have been a couple of studies that have been done, one recently from Ontario, where they were just testing the bulk tanks to see where they could find positive animals in herds, just in general, and not specific animals like we were. And so they found 82% of herds. So they tested really a lot of bulk tanks, and producers weren't paying, so they didn't really have to opt in or out or whatever. But at the same time, in the Maritimes, so that's on the east coast of Canada, they also did a similar study and found 90% of herds had at least one animal with leukosis. Within herd prevalence, so for those herds that test positive for leukosis, it's about 40% of their animals that are being tested are testing positive for leukosis. So that's pretty high, I think, pretty alarming, and I think good evidence for why we would want to make permanent like genetic gains in improving resistance to leukosis. Okay, in terms of heritability, so we estimated with our animal model about 10%, and for with the single-step GBLUP, 7%. The difference here is just the single-step GBLUP was able to partition the variances better, and so that's why there's that small difference there. And when we compared the top 10 sires, so we were looking at their EBVs or their ranking for leukosis resistance. So our top 10 sires, the more resistant animals, 29% of their daughters tested positive for leukosis on average, while for the bottom 10% of sires, 46% of their daughters tested positive for leukosis. So we do see a difference here. We also looked, as I said, at genetic correlations between different traits that are also really important and already being selected on in Canada. So milk, fat, protein, somatic cell. We also have mastitis resistance, herd life, which would be related to like longevity, metabolic disease, daughter fertility, and then we have our two indexes, which kind of include all of these things together. So you can see that there are favorable correlations for all of these traits. And if we look at the estimated breeding values over time from 2000 to 2015, we can see the general trend is that they have also been increasing at the same time. So already, without directly selecting for leukosis, this has already been happening anyways. And this has been seen also in the US study as well. The magnitude of the correlations weren't as high as what we found here, but they were also all favorable correlations as well. And it might have to do, again, with what I mentioned before about this idea that like you're going to keep animals in your herd that are producing milk and have good fertility, and so maybe these animals are already just being culled already without producers really realizing. Okay, so we did a GWAS for leukosis resistance. Basically, we just back-solved from our single-step GBLUP and estimated the effects for all of the SNPs. And so we only So we looked at chromosome level false discovery rate. And so we only had this one peak that was significant for our results. And this is where we have the major histocompatibility complex. So we have found 3 candidate genes, all kind of located in the major histocompatibility complex. Sometimes it can be a bit suspicious to get this kind of result, but we have also seen in other studies that they also found associations with the major histocompatibility complex with leukosis resistance in cattle, in other breeds of cattle. But Renee and I did had a lot of discussion on this topic and what can we do to make this better, because, well, our inflation factor is not the best, and working with a binary trait can be quite challenging. So we were talking about, it might be easier if we had a trait like proviral load. In leukosis resistance, or basically if an animal is infected with leukosis, it will have this proviral load, and those with the low proviral load are shedding less and spreading the disease less, and those with a higher are sharing it a lot more. So it might be beneficial to look at that instead of this binary trait. So here it is, 87% of herds were infected of those herds that were testing for leukosis. 39% of cows infected on farms with leukosis of those that were testing positive, and then our heritability was between 7 and 10%, and we had favorable genetic correlations with other traits. And our candidate genes were located in the major histocompatibility complex. So I think our conclusions from this are that it's definitely a candidate for including in probably like a health index or a resiliency index for the Canadian dairy industry. So this next study is a bit different from what I normally do. And this was from the master's thesis of Aisha that I was working with. And so Evan always asks me, what do we do after a GWAS? And this is the answer to this question, what do we do after a GWAS? And so we luckily had a really great collaboration with Professor Caro at the University of Guelph and his postdocs and research associate, Umesh and Milani. So Johne's disease is caused by a bacteria, and it's, I'm just going to call it MAP bacteria because that's quite a mouthful. It's contagious, it's chronic, it's fatal. Clinical signs appear more than 2 years after infection, and no commercial vaccine is available in Canada. My understanding of this is because there have been vaccines developed. As all vaccines, they're not 100% effective, but I believe that those animals inoculated with the vaccine will test positive, and so that's not very good for managing the disease. So, yes, so we do have an ELISA test, milk ELISA test for this as well. Unlike the leukosis test, which had a 99% sensitivity, we only have a 30% sensitivity for Johne's disease, which is really bad. So we have a lot of false negatives that are occurring when testing animals. And let's see, maybe I can explain a bit. So most of the infection is occurring in the colostrum when the calves are taking up the colostrum. So basically, it can be shed into the milk. The calf will ingest it, and then it can be 2 years until or more before this calf starts showing signs. So at first they'll be subclinical non-shedders, but then they become subclinical shedders, so they're spreading the disease, but you're not going to really notice that they have it. And then finally, clinical shedder, and that's where you'll start to see the chronic wasting. So what's happening is that the infection occurs in the intestine, and it's an initial infection of macrophages, and eventually it just goes from there. Okay, so the prevalence in Ontario, that's the province where University of Guelph is from, is the herd level is 26.2 to 27.2%. That's an estimate, and within herd, it's 2.3 to 4.2%. So before this study and leading up to it, there was an estimate of heritability. It was 6%, so quite low, I think, as we expect with most disease resistance traits. They also did a GWAS as well, and that's where they saw that this variation in this gene called IL-10R alpha was associated with variation in resistance to Johne's disease. So IL-10 and IL-10R alpha are upregulated in the host and during infection to MAP bacteria, and it's actually believed that this upregulation is caused by the MAP bacteria because it decreases the inflammation and makes the host more easy to infect. At the same time, it's also believed that MAP bacteria is also affecting the metabolism for the same reason, basically to make this a better environment for it to infect. So I have this nice little diagram here of IL-10, and then you have your subunits. So IL-10R alpha, there are two of those, and they make up two of the subunits in the receptor. And so basically, having more IL-10 and having more IL-10R alpha or receptors for IL-10 results in this activation of anti-inflammatory response. So we know this information already, but we want to look a bit further into actually what is happening, and are these kind of speculations, are they actually correct? So we wanted to examine the effect of live MAP bacteria on IL-10R alpha knockout bovine memory epithelial cells. I'm going to call those MAC T cells, by examining differentially expressed genes. And so we had 4 groups that we compared between looking at expressed genes. So we had our wild type MAC T cells that are not infected. We have our wild type MAC T cells that are infected. And then we have our knockout, the IL-10R alpha knockout MAC T cells that are not infected, and our knockout that are infected. And so we just looked at these 4 comparisons that I've illustrated with the lines here. So I'm going to be super brief on this part because I guess the other co-authors are the experts here. But this is what they tell me happened. They knocked out IL-10R alpha in the MAC T cell line using CRISPR-Cas9. They infected the cell lines with MAP bacteria. Experiments were carried out in quadruplicate at independent times, and the extraction and sequencing of mRNA then happened. And then there was quality control and identification of differentially expressed genes between the 4 groups that I showed already. So here are the number of differentially expressed genes for each of our comparisons. I think it's interesting to show that with our knockout versus our knockout that was infected, we have a lower number of differentially expressed genes than the other comparisons. This could be just because the only difference here is the MAP infection, and maybe there's not so much of a difference in the response to that when IL-10R alpha is being knocked out. Our 3 major processes were cellular process, biological regulation, and metabolic process. and so nothing to do with immunology, but apparently I'm told that this is actually quite common in these cell line studies. So what we did was then we just focused on immunology-related pathways and genes. But before I get into that, I did mention that the metabolic response is changed due to, we believe, a MAP infection. So we did look a bit into these metabolic response, and we found enrichments in pathways including cholesterol metabolism, retinol metabolism, fat digestion and absorption, and peroxisome proliferator activated receptor signaling. And so studies have shown that lipid metabolism and cholesterol pathways are affected by MAP infection to facilitate survival, as I said, and to establish infection and persistence within these intracellular compartments. So it kind of makes sense that we might be seeing these, especially in those comparisons where we didn't have the knockouts, because sometimes also metabolic response is changed in the knockouts. And then briefly just looking at our immune response in our 4 comparisons. So in our wild type versus wild type infected, so we're just looking at a regular infection here. We see an upregulation of some gene that is involved kind of in a similar pathway as IL-10 or alpha, and downregulation again of another inflammatory cytokine that's related as well. In our wild type versus our knockout, so no infection here, we again see some upregulation and downregulation in these different pathways, especially this pro-inflammatory pathways. And in our wild type that were infected and our knockout that were infected, again, we see these enrichments in these pathways that are related to our IL-10 or alpha. And of course, we saw this decrease in IL-10 or alpha because, well, we knocked it out. And then I think the really interesting comparison that we made here was between the knockout and the infected knockout. And again, we see a lot of upregulation of inflammation mediated by chemokine and cytokine signaling. So it really seems like IL-10 or alpha is of course related to inflammation, and that this change in response happens during infection, and that this new change happens when IL-10 or alpha is not there. Right, and then CCL20, which is in this group of genes that I was just talking about, has also been associated with inflammatory human bowel disease as well, so kind of getting related traits here in humans. So what does this tell us? I think it's support for IL-10 or alpha's role in MAP infection and pathogenesis in dairy cattle, and the inflammation is mediated by chemokine and cytokine signaling pathway, but also a couple of other pathways as well related to the immune response. And our results could help with the development of therapeutics, because maybe this is something that we can look further into with, like, maybe reducing this kind of inflammatory response, or actually increasing the inflammatory response, doing something with it. Then the immune response to Johne's disease and progression of MAP infection, I think is very complex, and we now have insight into other genes and pathways that are involved, and we can start thinking about those as well. And then just as a final conclusion, again, I think Leukosis and Johne's disease, as I've shown, their prevalence is surprisingly, actually for me, quite high in Canada. And I think that we need to use all the tools that we have available to us to try and reduce the prevalence of these diseases in Canada. And I think genomics clearly gives us a path forward with this. And yeah, this was my work at the University of Guelph. And of course, I'm here now. So feel free to come and talk to me anytime. Send me an email or whatever. And I'm happy to take any questions or comments that you have. Yeah.
Thank you, Chrissy. The question in the room, and just popping it in the chat.
Chrissy,
Yeah.
so for the first study, all those correlations with some of the traits that might have higher heritability.
Yes.
Is there any very nice proxy trait that would serve purpose for breeding purposes then, instead of doing additional analysis? If they cost farmer money, I mean.
Yeah, so I think like the other thing that we have to be worried about when having an index is that you want to put in your most important traits, right? Because you start to kind of lose progress on other traits if you have like a million traits in there. So I think that the work that's been done on leukosis resistance should now be kind of, I think this should be discussed further by the people who actually make these breeding decisions, because it might be that we just say, well, it seems that longevity seems to do the trick well enough for us, and maybe we don't add something new to our indexes.
Okay, thank you. And then second question is, some 7-8 years ago I was in seminar from somebody from Guelph, and then they were talking about the selecting cows for superior immune response. Is this kind of things part of that? Or is superior immune response something more general?
So I think you're referring to the CMEX markets
Yes.
and Immunity Plus.
Yes.
And so I've asked around a bit about this because I'm curious, and it's also patented, so they don't really explain too much. But my understanding is it's also that they're selecting on something kind of within the MHC region.
Sure.
But this work is not associated with those guys that have been working on Immunity Plus. But I would be curious to see how they are related to each other.
Okay, thank you.
Yep.
The effect of the herd size for the first trait, as in, if you have a larger herd, there's less chance of a lot of, or the other way around, for the disease to really spread?
Not that I'm aware of. I don't think there was, yeah.
About that and what you said also on the level of infection, did you look into the infectivity? So you said that there is probably a test that can tell you if an animal has a high level of infectivity, and then they're more likely to be a spreader, or they are less likely to be a spreader if they have a low level of infectivity. So how good is that test, and could you use that instead of using the ELISA? Because what you want to reduce is, well, what you want to increase is not only the resistance, but also you want to reduce the infectivity level.
Yeah, exactly. So I think, if I understand correctly, I believe that it's quite an accurate test of proviral loads, because it's just counting, right, the viral pieces of whatever, as an RNA or something. But this is in blood. So it kind of takes away the easiness of the milk ELISA tests, right? Because then you'd have to go and actually draw blood and do those tests. And yeah, it's a lot extra. Yeah. Mm-hmm.
I have two questions.
Sure.
In your model, it's only the female you take into the account after calving, or male are also included in your GBAS? So the sex is considered, or was it checked for certain specificity if they have more prevalence in male or female?
So with the milk ELISA tests, they're
Yeah.
milk tests. So none of the males have any tests. So we have no data on whether males have leukosis or not. but we do still include them with like the pedigree information and genomic information, right? So that we can still estimate their breeding values. So they're included, but we wouldn't be able to estimate any kind of effective sex.
Okay.
Yep.
Do you also have temporal data on those cows which are infected? It means like you have a data of like 3 years exactly on same herd, and some are infected, some are not, and you see the gradual increase and decrease in your phenotypic values, or like they are infected or not, because you said once they are infected, they are always have reduced. So did anybody do this temporal study on them?
Yeah, so the tricky thing with this data is that because it's a producer opts in and pays for the data themselves, it's not necessarily that they test their animals every single year or like for 5 years in a row or anything. They may have just tested them once or that's it. So it's really in the producer's hands, and we're just looking back at the data that's available. So for that reason, we weren't able to look at it over time in herds.
And if the money allowed, would they allow to do it?
Yeah, that's an interesting question. If producers would be interested in looking at that over time, if somebody else was paying for it, I'm sure that they would be more than happy. Because again, like I said, it's just a routine in the milk test, right? So I don't see why not.
But are you saying no one has opted in to get a time course measurement? Or?
It could be that some of the herds have looked at like many years, but not enough for us to actually do some kind of statistical analysis. Yep. Are there any questions online?
There is one.
It's me.
Ah.
I'm telling them if you have questions.
Okay.
Can I ask another question about
Sure.
your GWAS on the leukosis? So you had that not very good lambda value. So
Yeah.
did you try fitting principal components or something, like the kinds of things that we often do to kind of combat that kind of problem?
But isn't it like the low lambda value that you don't have issues with stratification, and it's the high one that you do, that you should fit the principal components?
So what does it indicate then to have a particular? You think it's just to do with it being a binary trait?
Yeah, I think that could be it, but I'm just not really sure. Yeah. Okay. Well, thank you again, everyone, and I think that's it. No more questions? Thanks.